The beef cattle industry is the last major livestock species that spends the majority of their life outside. And so I think the beef cattle industry is also a lot more heterogeneous than both in terms of environment and management than a lot of our other livestock production systems, especially in the cow-calf sector. I think this is the industry's biggest strength, but can also be its biggest weakness, because it means that cattle are subject to a very wide array of stressors, and the ideal phenotype can vary by environment. And so to that end, there's been some interest in augmenting the current systems that we use to evaluate genetic merit at a national level with more environmentally aware predictions. And there's two main strategies that we could use to do that. The first is through the development of novel phenotypes, which is going to be one of the things that I talk about today in the first part of my presentation. And then secondly, modification of methodologies that already exist to evaluate phenotypes in a new way, which will be the second part of my presentation. Sorry. There we go. So like I said, the first two chapters I'm going to discuss explore that first strategy using a novel phenotype called hair shedding score, and are kind of different sides of the same project. The first one I started as an intern at the American Angus Association, which is the largest beef cattle breed association here in the US. And it focuses more on the quantitative genetics of hair shedding score and development of an EPD for producers to use to select more heat tolerant cattle. The second part was completed here at the University of Missouri and focuses more on the functional aspects of hair shedding score and identifying genetic loci that contribute to variation in hair shedding score. The third project was also completed at Angus. I was very lucky to be very busy while I was there. And it focuses on that second strategy, using new methodologies or looking at data in a different way. Here we're looking at genotype-by-environment interactions for weaning weight. And so I'm going to talk about the first two chapters pretty interchangeably, but I'll try to refer to the AGI or the Angus data with this green color font, and then the Mizzou project or data that came from that project in blue. So nearly all mammals replace their coat either annually or biannually as a response to seasonal variation. And the mechanisms that control this coat change are largely shared between species and tied pretty closely to seasonal reproduction as well as seasonal growth. And so the underlying biology that is associated with those seasonal phenotypes is pretty well understood to be controlled by changes in day length, but very little research on the genetic basis of seasonal coat change has been undertaken to date. And so we don't really have a good understanding of how genetic variation affects coat shedding or an animal's ability to respond to environmental cues and alter their phenotype accordingly. And seasonal coat change is less strongly tied to fitness in domestic species like cattle, but can still have huge production impacts. Heat stress is one of the biggest contributors to economic loss in the beef industry, accounting for over $500 million a year, particularly in the cow-calf sector. And so in early summer, cattle that shed their winter coat later will have to partition more energy towards thermoregulation that could have gone towards growth or parental care. And so anecdotally, we'll often see those late shedders hanging out in the pond or under in shade while their peers are out grazing. A group in Australia noticed this back in the 60s and developed a system to quantify differences between individuals in hair shedding score and other coat characteristics. Other systems were independently developed, including this 5-point scale that we're using today, where 5 is 0% of the winter coat shed, so this really hairy cow here at the top, and 1 is 100% winter coat shed, completely slick. And there's quite a bit of variation in the pattern of that hair shedding as we advance from a 5 to a 1. But as a general rule, mammals shed their winter coat from front to back and top to bottom. And so if you compare the hair shedding score 4 to a hair shedding score 3, we can see that the 4, she's lost really only the hair around her neck, whereas the 3 is starting to lose along her top line and towards her shoulders. And then similarly comparing a hair shedding score 3 to a 2, We can see that the only hair that the hair shading score 2 is holding on to is around her flanks and on her belly. And so our goal for this project was overall to identify cattle that are better hair shading score, have better genetic meta for hair shading score, and create selection tools for beef cattle producers to use. And kind of going into that, we were concerned about what extrinsic factors affect variation in hair shading score so that we can correctly model them in our predictions, and also so we can compare these effects to what we know in other species. Second, we were concerned with understanding how genomic variation contributes to phenotypic variation in hair shedding. And this has implications both for our prediction of genetic merit, as well as our understanding of the underlying biology and genetics of hair shading score or of seasonal coat change. And since we know that this is so closely related to seasonal coat change in other species, and it's very highly conserved, we know that this could have implications for our understanding of seasonal biology in general. With that, we sought here at Mizzou to generate a large multi-breed data set of hair shedding scores recorded in different environments and at different time points in animals' life. And so we enrolled producers in this project back in 2016, and basically in exchange for 3 years of hair shedding scores on their herd, we genotyped that producer's herd for free using this GGP F250 genotyping chip that we developed here. And then those genotypes were then transferred to the breeders, their respective breed associations, and incorporated into that breed association's genetic evaluation so that they essentially got free genomically enhanced EPDs for participation. And so all in all, we're close to 40,000 total hair shedding scores. And nearly all of the animals that we have phenotypes recorded on have been genotyped on that assay that I mentioned, and then imputed to higher density using this method that Troy Rowan developed. And so the American Angus Association has also been interested in hair shedding scoring for a while. Back in 2011, they funded several Angus Foundation projects to collect hair shedding scores on registered Angus cattle in the Southeast in the Fescue Belt. And so during my time at Angus, I basically brought the hair shedding scores on registered Angus cattle that we had on hand here and combined them with that legacy data. And based on that work, there's currently a research EPD available for producers to use to identify AI bulls that have high or low genetic merit for hair shedding score. And we recently published the paper describing the development of that genetic evaluation in GSE. And so in both halves of the project, this is the general model that I use to estimate genetic parameters and breeding values. And if you're more used to thinking on an EPD scale, a breeding value is just 2 times the EPD. And so we use this repeated records animal model to fit both an additive genetic effect, or the breeding value, and then a permanent environment effect, which I'll talk about later here in the next slide. We use a single step method to combine information from both genotyped and non-genotyped animals. And basically what that does is it blends the additive genetic relationship matrix with the genomic relationship matrix to take advantage of all information available. And then every model that I'm going to use in the hair shedding section is going to be just some slight variation of this model. And when we use a repeated records model, like I mentioned before, we can also estimate a permanent environment effect in addition to that additive genetic effect. And so what I mean when I say a permanent environment is anything that contributes to every record on an individual, but can't be passed on to offspring. So it's not part of the genetic merit. And when we have a permanent environment effect, we're able to estimate this repeatability measure, which is basically provides the upper bounds limit estimate for the heritability. When we look at the heritabilities and repeatabilities that we've found in the AGI data, as well as the Mizzou data, and the Mizzou data subset to purebred subsets, we find that the heritability is quite similar across all of these subsets, about 40% heritable, which is similar to estimates reported using Pedigree relatedness back in 2011. We also find that permanent environment contributes about 7% on average to total phenotypic variation. When we stratify our breeding values from the Mizzou dataset in that model that I mentioned, we find that variation largely overlaps between breeds. However, there is some tendency for breeds that were recently selected for the show ring, so here Maine-Anjou and Shorthorn, to have less desirable EBVs, which we think might make sense. Further, breeds with Bos indicus influence, so here Brangus and Green, tend to have more desirable EBVs, which again makes sense since we know that Bos indicus cattle have been selected in their evolutionary history for heat tolerance. So Charolais cattle in the US, almost all of them were imported through Mexico. And so during that time, they were interbred with the Brahman cattle there. And as a result, nearly all Charolais cattle currently have at least some low-lying percentage of Bos indicus ancestry floating around. So we think that might be part of the reason why we have Charolais ranks third in terms of the most desirable on average breeding value. I'm not super sure what's going on with Gelby here, but I think it's kind of interesting. And if anyone has any ideas, I would be glad to hear them. So one of the first things we were concerned about in developing the AGI breeding value was how grazing endophyte infected fescue affects variation in hair shedding score. So if you're unfamiliar, tall fescue is the most prevalent, or one of the most prevalent forages here in the US, or here in the Midwest and the Southeast. And it often harbors this endophytic fungus that protects the plant, but to varying degrees causes side effects in livestock. And these symptoms are collectively referred to as fescue toxicosis. And so one of the symptoms of fescue toxicosis is vasoconstriction and disruption of follicle development, which reduces an animal's ability to shed their winter coat and exacerbates heat stress. And so we weren't sure if the mechanisms or controlling hair shedding score on versus off endophyte infected fescue were the same, and we wanted to be able to model them correctly so that we don't introduce bias unnecessarily. But when we fit a bivariate model where we treat records from animals that we know were grazing endophyte infected fescue as a different trait from those that we know that weren't, and we can estimate a genetic correlation between those two traits, we find that they're pretty highly correlated with very minimal re-ranking of breeding values. So I take that to mean that we can safely fit them as the same trait in a national cattle evaluation. However, we find a larger effect of permanent environment for cattle grazing endophyte infected fescue versus those that weren't, and therefore a higher repeatability. And so I think that this might be hinting at the impact of accrued physiological damage from fescue toxicosis on variation in hair shedding, since we know that it can cause long-term damage in the venous cavities and in other parts of the body. I think evidence for this is we have more evidence for this whenever we fit a fixed effect model where we treat all records as the same trait and just estimate an effect of grazing versus not grazing endophyte infected fescue. And when we do that, we find that on average, cattle that were not grazing endophyte infected fescue, grazing some other improved forage, have on average about a half a score more desirable hair shedding score. And so another consideration for implementing this in the national cattle evaluation was the effect of age, because we know that for some traits, the expression of a phenotype is not consistent across lifespan. And previous research in other animals regarding seasonal coat change and the effects of age suggests that the oldest animals and the youngest animals tend to shed later than their middle-aged peers. And it looks like we find the same kind of effect here in beef cattle. So as might be expected, 2- and 3-year-olds tend to have the highest or least desirable hair shedding scores, which makes sense when we consider that they're still growing and also experiencing the new stress of lactation. However, we find less of a disadvantage to those very old animals than what we see in other species. And when we compare the effects that we found in the AGI model to the Mizzou data set, that effective age among old cows is even lower. I think this might be a reflection of those very old cows in our data set being a selected sample. We know that most cows either die or leave the herd by 11 or 12 years of age. And so we think that these ones that have survived and are still being productive and late in their lifespan are those that are very well adapted to their environment. I think of this old cow result here, the low effective age compared to what we find in other species, could also be support for early summer hair shedding score as an important characteristic of longevity in heat stressed environments. So as I mentioned in my introduction, the biology that underlies seasonal coat change is largely shared between species. And so here on the right of the slide, we have the main hormonal pathway that regulates seasonal hair change. And so first, changes in day length or increasing day length at the beginning of the summer is sensed in the eye, which then forwards that signal on to the pineal gland. And the pineal gland is responsible for producing melatonin, which is inhibitory of prolactin. And as I mentioned, I think prolactin is the main hormone associated with initiation of hair shedding score, or hair shedding, seasonal coat change, sorry. So when day length increases, production of melatonin decreases and removes that block on prolactin, allowing the initiation of seasonal coat change. And so in some mammals, changing temperatures magnify the effects of day length to hasten molting further. However, the mechanisms that control temperature sensing are not very well understood in mammals, nor how they influence seasonal coat change. In beef cattle, the effects of temperature and day length have been independently demonstrated, but to our knowledge, not their interaction. And so in order to quantify their relative contributions to hair shedding score, we compared a series of models, including the effects of day length, temperature, or both. And so first what we did was infer the geographic coordinates of all of the herds in our project using the producer provided address. And then based on that address, we pulled down the day length and temperature information for every unique score date at that location and the 30 days prior to that score date. And then these values were then averaged and assigned to hair shedding scores, such that each hair shedding score had a summary value over the last 30 days for day length and a summary value for temperature over the last 30 days. And so for day length, we just subtracted the time at sunset from the time at sunrise. And then for temperature, we use the apparent high temperature. And apparent temperature, the feels-like temperature, differs from the direct temperature in that it just incorporates wind speed and humidity. And so day length and temperature are moderately correlated. And so when we fit our interaction, we center that variable. This model here is the base model, including both the effect of day length and temperature. But we fit 3 additional models, including just day length, just temperature, or day length, temperature, and day length multiplied by temperature for the interaction effect. And like I said, we centered that interaction effect. And so the model, including the interaction between day length and temperature, gave the best fit to our data. However, all of our models suggested that as day length or temperature increases, hair shedding score on average decreases. And the magnitude of the day length effect was larger, with nearly half a score decrease in hair shedding score with each hour increase in day length over the last 30 days. And this is in line with what we expect from other species. So to gather all of the results from these analyses that I've talked about up until now, examining the non-genetic factors that affect hair shedding score, give us a good start as we began to search for genomic loci impacting variation in hair shedding score. And so when we run these genome-wide association analyses, or GWAS, it's a little tricky to do with repeated records because there aren't many softwares that can both incorporate a permanent environment effect and handle the size of data that we had available to us. But we wanted to be able to take advantage of the increased prediction accuracy that comes from using repeated records. So we chose to deregress our breeding values and use those as a pseudo phenotype in these GWAS analyses. And so just a quick explanation of how to look at this plot. The numbers on the x-axis are chromosomes in the bovine genome, and each point in these chromosomes are physical positions in the chromosome. And so the number, or the value on the y-axis, is the significance of that point in the genome in causing a change in hair shedding score. Yes. So we find associations on nearly every chromosome, but I think particularly interesting to point out are these associations on chromosome 5, chromosome 13, 22, and 23. And so when we zoom in on that huge association that we found on chromosome 5, we can see that this large 10 megabase region probably contains 2 independent associations. We originally found the same peak, or the same association, in the AGI project, but we were unable to narrow it down to whether or not it was just one association or one massive, or 2 associations or one massive association, due to extensive linkage disequilibrium that comes from working with a purebred data set, as well as lower genotype density. So when we impute our data up to higher density, and we use a multi-breed data set to break up some of that long range LD, it gives us better resolution to say confidently that there's probably 2 associations going on here. This is a very gene-rich area, but I think particularly interesting are these CEP290 and KIT ligand genes located within the association. Because it turns out that both KIT ligand, or KIT-LG, and CEP290 are involved in ocular function. CEP290 playing a role in the production of one of the cell types that's involved in light sensing. And then KIT ligand and MITF, which we identified within our association on chromosome 22, are important master regulators during development. And MITF is actually regulated via KIT signaling. So among KIT ligand and MITF's many roles include maintenance of retinal epithelial cells, and mutations in both KIT ligand and MITF result in a number of well-documented retinal disease phenotypes. Additionally, when we look for QTL and gene ontology enrichments using the SNPs that we found to be significant in our GWAS, we find gene ontology enrichments associated with light perception and optic nerve misrouting. We also find evidence for variation at other parts of the pathway regulating seasonal coat change. So as I mentioned before, prolactin is the main hormone associated with the initiation of hair shedding. And mutations in the prolactin gene have well-documented associations with abnormal milk production and thermoregulation beyond its effect on coat change. Interestingly, we find several significant variants near this prolactin gene on chromosome 23. And kind of going along with that idea, we find gene ontology enrichments for photoperiodism. One of the other ways that seasonal coat change is regulated is indirectly via sex steroid hormones like estrogen and testosterone, and their impacts on prolactin. Among our ontology enrichments, we also found terms associated with testosterone regulation. So in addition to variation at multiple points in the pathway regulating seasonal coat change, we find quite a lot of evidence for effects of metabolism, including enrichments for growth hormone signaling and QTL associated with body weight. To be honest, I'm not super sure what to make of these results, but I think I have 3 possibilities in mind. So first, we know that day length can affect weight gain and nutrient partitioning in cattle. To my knowledge, there isn't a demonstrated relationship between seasonal coat change, growth, and photoperiodism, but that might have something to do with what's going on here. I think the second explanation is maybe the most parsimonious. It's just that growth is a super complex trait. And so if you throw a dart at a random place in the genome, you're probably going to hit a growth QTL. And there's also enrichment bias or annotation bias for growth QTLs. So maybe we're tagging something else or other things, but just picking up these growth QTL because they're so highly annotated. Finally, I think that it's possible that there might be some other link between metabolism and hair shedding, not related to day length. I think it's interesting to point out that we have many variants near and within this CDKAL1 gene in our second largest association on chromosome 23. And in humans, CDKAL1 has been repeatedly associated with susceptibility to type 2 diabetes, growth, and obesity. When we zoom in on that chromosome 23 association, we've seen that SNPs within CDKAL1 or actually within CDKAL1 make up most of our most significant SNPs in that association. And so as I mentioned before, the SNPs that we found near prolactin were also part of this peak on chromosome 23. So it's possible that those prolactin hits were just driven by linkage to the CDKAL1 association, or that there's more than one association driving this signal. I think in the future, bringing this conditional joint analysis, where we condition on our significant SNPs in order to determine the number of independent associations within a peak, would help us refine what's going on in this chromosome 23 association and elsewhere. So finally, we wanted to evaluate hair shedding score as an actual indicator of one measure of dam productivity weaning weight. So as I mentioned previously, heat stress accounts for a huge proportion of the economic losses in the beef industry every year. And one of the ways that heat stress impacts the cow-calf sector is indirectly via reduced dam productivity and that's impact on calf weaning weight. And so what I mean when I say weaning weight is the weight of a calf at about 7 months of age when it's weaned off of its dam. And I just wanted to show this equation to point out that when we calculate breeding values or genetic merit for weaning weight, we fit both a direct effect of the calf, so the impact of the calf's genetics on its propensity for growth, as well as a maternal effect of the dam. So basically the effect of the dam on its calf's weaning weight beyond its additive contribution to that calf's genetics. And so maternal genetic effects can arise in a number of ways. Most often we talk about them most frequently in the context of milk production, but they can also arise via behavior and mothering ability. And so the relationship between weaning weight and hair shedding has been previously demonstrated, but it's always been looking at the direct effect of weaning weight, which isn't really the relationship that we're interested in. And since we're more interested in dam performance, we fit this bivariate model where we fit the maternal effect of weaning weight and hair shedding score. And when we calculate a genetic correlation between those two traits, we find that it's pretty consistently, regardless of environment, somewhere between 20 to 28% negatively correlated. However, whenever we look at the maternal effect of weaning weight and its genetic correlation to the direct effect of weaning weight, we find that there's a difference in the magnitude of that effect based on environment. So this negative genetic correlation between the maternal and direct effects of weaning weight consistently comes up, and it's kind of puzzled researchers for many years. So some people have suggested that it's an artifact of the statistical models that we use, and others have suggested that it's reflective of real biology and genetic antagonism between growth and milk production. And I think that our results here might give more evidence for that. So I think that what we might be picking up is the effects of grazing toxic fescue on nutrient partitioning in the dam and creating more of a genetic antagonism between that maternal and direct effect. So that was kind of an interesting result that didn't really have to do directly with weaning weight or with hair shedding score, but was interesting to us. And so wrapping up the hair shedding score section, I think the biggest takeaways are that Hershing score is moderately heritable and a good candidate for an EPD. Besides its heritability, it's a very low input phenotype for producers to record, and the fact that it's a repeated record traits lends itself to accuracy. I think in the future, it'd be helpful to see Hershing score combined with other high dimensional or gold standard measurements of heat stress in order to create even more robust predictions. We find similar effects of extrinsic or non-genetic variables as in other species, which helped guide us in how we modeled Hershing score, as well as informed our understanding of function. In our GWAS, we found evidence for variation at various points in that main pathway that regulates Hershing score, as well as variation in metabolism. In the future, before we submit this final Mizzou paper, I think it'd be interesting to try this G/E GWAS analysis, where we fit an interaction effect between a variant or a SNP and a environmental gradient like day length or temperature, such that we can identify variants with differing contributions to variation in phenotype, depending where on that gradient they're expressed. And so circling back to this outline that I talked about in the introduction, we focused on the development of a novel phenotype. And the second way that we could focus on creating strategies for environmentally aware predictions is through modification of existing methodologies to look at data in a new way. And that's what we're going to talk about for the rest of the presentation. And again, this is a project that I started at the American Angus Association. We focused on the evaluating the degree to which genotype-by-environment interactions impact growth phenotype in American Angus cattle, and exploring if incorporating these genotype-by-environment interactions into the models that we currently use to evaluate genetic merit might one day be worth pursuing. So I think we've probably all seen this little equation. It's probably the quantitative genetics equivalent of the molecular biologist's central dogma. And so while that may be, it doesn't tell the whole story, because a lot of what contributes to phenotypic variation are these genotype-by-environment interactions. And so in addition to the heterogeneity in the beef cattle industry that I discussed in my introductory slides, there's also widespread use of artificial insemination, or AI. But most AI bulls are raised in much more favorable conditions than their progeny, which creates the potential for these G/E or genotype-by-environment interactions, whereby the effect of a genotype differs depending on the environment in which it is expressed. So based on that, we sought to quantify both the phenotypic and genetic differences in growth across the US. We had these three questions. So first off, how much does actual cattle growth phenotypes differ between environmental regions? How much of that difference is due to differences in management in the environment versus genotype? And then finally, how much ranking of genotypes do we find across regions? And are there any variants that significantly interact with the environment? So again, this is the same slide that I talked about in my hair shedding section. And again, I'm just bringing it up to point out that we fit both a direct and maternal effect when we calculate weaning weight, which is the phenotype I'm going to be focusing on for the rest of my presentation. And we're looking at weaning weight through the lens of these discrete ecoregions created by my colleague, Troy Rowan. And so what Troy did at the beginning of his PhD was gather temperature, precipitation, and elevation data over the last 30 years for every 1 kilometer square of the United States. And then using k-means clustering, he assigned all the United States to one of these 9 discrete climate regions. And so I'll talk a little bit about all the regions, but the three I'm going to focus on the most are the high plains, which is where most of our seed stock operations are located in the Angus breed. And this means that most cattle can trace at least some degree of their recent ancestry back here. The Southeast, which we a priori expect to be the most stressful region due to high heat and humidity. And then the Fescue belt, which is unique in that it's a temperate environment, and we have many seed stock or breeding operations located here. But this endemic fescue toxicosis or endemic forage produces a toxin with an effect on cattle and makes it a very stressful place for weaning weight. So as I mentioned earlier, the widespread use of artificial insemination creates a unique system for us to look at the performance of genotypes across multiple environments because we have these huge half-sibling families. So on this point, each dot is a bull with at least 50 calves in each of the high plains, which again is where most of our seed stock operations are located, and one of the 6 comparison regions. So on the x-axis, we have the median weaning weight of that bull's calves in the high plains. And then on the y-axis, we have the median weaning weight of that bull's calves in a comparison region. And so in the absence of G by E, we'd expect all of these points to fall along the diagonal line, and many of them do. However, we find a lot of variation along that line. And there are some huge deviations. So colors, or the points are colored by the difference in the weaning weight of that bull's calves in the high plains minus his calves' weight in the comparison region. And so the further down and to the right of this line you are, the worse your calves do in the comparison region compared to your calves in the high plain. This is often like 100 pounds or more difference between bull performances across regions. And so in conclusion, this gives us phenotypic evidence for possible G by E and prompts us to explore further. So now that we've established that there are phenotypic differences across these discrete regions, the next step is to quantify the contribution of the environment to phenotype. The way that we typically do this in animal breeding is using contemporary groups, which is basically a fixed effect that we include when we're estimating genetic merit so that the environment doesn't affect our estimate of the breeding value. And so the way that contemporary groups are implemented for weaning weight at the Angus Association are these farmer-reported management groups, where calves have to have been managed in the same way, born within several months of each other, and the same sex. And so by solving the effect of contemporary group in these linear mixed models that I mentioned previously, we can quantify how much the environment helps or hurts the phenotype with that contemporary group solution. And when we stratify the contemporary group solutions that we get from the national scale cattle evaluation by region, we're able to get a picture of how stressful a region is. And so here in green, we have the high plains, and it's the highest ranked region of our regions in terms of contemporary group solution. And it has a 50 pounds higher contemporary group solution on average than the median contemporary group solution in the Southeast. And again, these results give us expectations for which regions we think are going to be the most stressful and most likely to be at risk for G by E. And the final step is to parse out what variation in phenotype, if any, can be attributed to G by E. The way we most often do this in animal breeding is using these random regression models, whereby we regress a genotype or an individual's performance on a continuous variable, like temperature or disease incidence, such that each genotype is fit to its own unique curve. Another way we can do this is using these multivariate models, where we treat observations on the same variable made under different conditions as separate traits and estimate a genetic correlation between them. And that genetic correlation is interpreted as the degree of G by E and re-ranking of breeding values between environments, or the two environments. And typically the threshold that we've used in the literature is this 0.8 threshold for very different environments suggested by Falconer back in the 50s. And again, we think that since we used our K-mean clustering of multiple variables to create these environmental regions, we're better able to capture stressors that haven't been explicitly measured through the combination of those variables. So I applied these in multivariate models using the high plains as an anchor region. And so basically for each of the high plains and the other regions, I sampled 100,000 total records by zip code. including only zip codes with at least 10 years of data, which I think helped us to have more accurate calculation of maternal effects and a maternal permanent environment effect. And then what we did is we calculated our variance components and genetics correlations in 10 bootstrapped iterations with resampling per region, such that we had 60 total analyses. And so when we look at the direct effective weaning weight and the results across those iterations, we find that the mean genetic correlation between the high plains and the comparison region for the direct effective weaning weight falls around the mid-80s, above that threshold that we've found in the literature. And I should also point out that here, this in records column is the pool of samples that we had to sample our 100,000 sample iterations from. There's a pretty wide range of genetic correlation estimates between these 10 iterations, but to me this suggests, based on the mean genetic correlations across those 10 iterations, that there isn't as much evidence for genotype-by-environment interactions in the direct effective weaning weight. However, when we look at the maternal effective weaning weight, that range of estimates across iterations is even larger, and the mean genetic correlation tends to be lower, especially here in the Southeast and the Fescue belt, which, if you recall, were those two lowest ranked regions based on contemporary group solutions. And the Southeast even falls below that 0.8 threshold that we talked about. So I think that the variation in genetic correlations that we find between regions and within a region between iterations is probably due to two types of sampling variants. The first one I'm going to talk about is cryptogenic variation, which is basically variation that contributes to, or genetic variants that contribute to variation in a phenotype in some environments, but not others. I think one good example we can use to think about this is plane of nutrition. So there are likely segregating variants that affect milk production when a dam is on a low plane of nutrition or stressed, but not when her nutritional needs are met. And so within the same ecoregion, we could be sampling low nutrition versus high nutrition between cohorts. So yeah, so the iteration containing many animals on a low plane of nutrition, those would have a higher estimated genetic variance due to the expression of typically cryptic staining variation. I think another good example of things that could potentially cause cryptic genetic variation to be expressed in one environment but not the other is calving season. So we know that heat stress places, fall calving is likely to be less stressful because of the dam not having to experience the heat of summer while she's raising her calf. When we look at the distribution of calving dates across our region, stratified by region, we find that it's bimodal in 4 of our regions. And so it's possible that from iteration to iteration, we could be sampling more or less of one calving season, changing which variants affect that phenotype. And again, I think this idea could be expanded to other environmental variables like pathogen prevalence and fescue toxicosis. So as I already discussed, fescue is a very prominent forage here in the Midwest. We could be sampling breeders that are grazing Endophyte infected fescue versus other improved forages. Another simpler way that sampling variants could be affecting our estimates is through sampling pedigree relatedness. So the method that we're using relies on having half siblings or pedigree relatedness between our two regions, between the high plains and the comparison region. And our variance component estimation was based on pedigree relatedness, not genomic relatedness. So recent work has shown that the estimates between between variance components estimated using a genomic relatedness matrix and a pedigree relatedness matrix should be similar. But this might not extend to our method because we didn't do anything to explicitly filter for relatedness between individuals. So it's possible that we have differences in the sampling of pedigree relatedness between iterations. And then finally, we began to answer the question of whether the architecture controlling weaning weight differed between regions. And we used a single-step GWAS method that combines both genotyped and non-genotyped animals, similar to the method that I mentioned in the hair shedding part. And basically, we calculate breeding values and are able to back solve for SNP effects and p-values for those SNP effects. And so we sampled animals in the same way that we did in the variance component estimation section, such that we ended up with about 10,000 genotype samples per each of the 3 regions that we decided to look at. The high plains, because like I said, it's where most of our seed stock operations are, and it was the anchor in those variance components analyses. In the southeast and the Fescue belt, based on our results from the contemporary group analyses and the variance components analyses. So when we look at the direct effect of weaning weight, we find mostly known growth QTL with very few differences between regions, including these huge peaks on chromosomes 7 and 20, which have been annotated many times as growth QTL. When we look at the maternal effect of weaning weight, we find more striking differences between regions. There's only one significant variant in the high plains, but we find quite a bit of variation in the variants that reach significance in the southeast and the Fescue belt. So unfortunately, we were limited to data at a fairly low genotype density, which I don't think gave us very good resolution to connect these variants to candidate functions. But I do think that this research, or these results, motivate further research at higher density. So basically, in conclusion, I think that we find less evidence for G/E and the direct effect of weaning weight than in the maternal effect of weaning weight. However, when you think about this in the context of a large-scale national cattle evaluation with millions of animals, even small degrees of re-ranking can be a huge upset. I think that's based on that. Our results motivate further exploration of G/E and weaning weight and other growth traits. I think that a lot of the variation that we saw between regions and between iterations within the same region in the genetic correlations is due to 2 types of sampling variants, sampling of cryptogenic variation and pedigree relatedness. And then, so finally, to wrap up, today I talked about 2 strategies to generate environmentally aware predictions. First, using a novel phenotype hair shedding score, and then secondly, modifying existing methodologies to identify G/E in weaning weight. Wrapping up, I really want to thank all of the breed associations that participated in this project. There are so many more people on here than, or that helped out than are on this list, but I think that these are the people that I just interacted with directly. And I know that more than one of these people probably dreaded getting emails from me, so thank you for always being nice and helpful as we worked on the data transfer part and the genotyping side of this project. I really appreciate it. It's been super cool to get to know so many people. I also want to thank all of the work, or thank all of the folks at the MU Extension and Research Farms, particularly Southwest and Thompson, and Eldon, who has been super helpful over the years, and actually was one of the people who independently developed a hair shedding scoring system a while ago. And it's always great to see you guys. I especially want to thank Stoney and Charles, because there's been a couple times where it's just been really hard going out to score cows at Thompson, and there was one time that I forgot my boots and it was bad. And so Stoney and Charles drove me around on a 4-wheeler and were very nice and very helpful and are always great. And I also want to thank everyone in the MU team and on my committee. I want to point out a few people here. Thank you to the Rowan family for being my second family while I was away from Texas and didn't get to see my family. It's been wonderful to be able to see you all the time. And then Troy, obviously, for being so wonderful and helpful, I don't know, keeping me sane. And Lauren for being my biggest hype man. I just really, really appreciate you. And my beautiful dogs, who are the cutest dogs in the entire world. And with that, I can open up to questions.
Thanks, Harley. Great job. So feel free to just unmute yourself and ask the question, or you can type it into the chat box and I'll read it out for the group.
Hey Harley, this is Dr. Lucy.
How's it going?
Can you hear me?
I can hear you.
So super good, really enjoyed that.
Thank you.
I had a couple of questions, like what do we know about hair shedding in Holstein? You know, we talk a ton about heat stress in lactating cows, because is similar work already been done or in the process of being done in Holstein?
Yes. What I know the most about in Holsteins was the project looking at there's these super hairy cows that have a mutation in prolactin, and it causes this weird like hairy phenotype and weird bad milk production. And even when you shave them, they still have thermoregulatory issues. I know what also comes to mind is not a hair shedding. I think it's important to distinguish hair shedding from coat characteristics, though.
Okay.
Different things, right? But a lot of work I know has been done with introgressing the slick phenotype into Holsteins. But I'm not super as familiar as with work looking at hair shedding scores in Holsteins.
Okay.
I think my intuition is that you'd find less variation, but I don't know if that's actually true in Holsteins and in dairy cattle.
I've never really seen similar work done, or maybe we just don't concern ourselves in the dairy with the shedding as much, or I don't know. But I had another question, and I think I just wasn't listening very carefully. You mentioned that one of the things that you talked about, metabolism, was one of the things that was coming up, and I probably wasn't listening carefully, but wouldn't you think that would have something to do with the fact that if you have a more metabolically active cow producing metabolic heat, that would influence its shedding? I mean, or
yeah, I think that's entirely possible. I honestly am not entirely sure what's going on there, but I think that makes sense. That's one thing Jared and I were talking about yesterday, is that maybe just fat cows shed better because they're at a higher, or able to reach a higher plane of nutrition easier, or more easily. So that's something to think about too. I hadn't thought about that.
Yeah, that's cool. It's interesting. Enjoyed that.
Well, thank you.
Harley, this is Tom McFadden. Not to be the dairy guys, but I just wanted to jump on and follow that. So it's interesting that a lot of your candidates arising from the GWAS, to me, were sort of big central regulation.
Yes, I thought that was very strange too.
Points. And so, do you have any other phenotypes that you tracked that might also be indicative? For example, age at first puberty, or things like that might also be light sensitive?
Yes. So age at first puberty has actually been shown in beef cattle and in dairy cattle to be influenced in part by, or the age at puberty, by photoperiod. Cows, I mean, obviously aren't strictly seasonal breeders, but there are various effects known for photoperiod in fertility. I don't talk about it here, but I also get some QTL associated with conception rate and non-return rate. But I didn't include them, but I think that's very interesting to think about.
Thanks, nice job.
Thank you.
Can I jump back in really quickly, Harley?
Sure.
Sorry, and then on your prolactin, I was looking at your prolactin gene, yeah?
Mm-hmm.
And I noticed you had a few QTL in there, and I'm assuming prolactin is probably about the same size as growth hormone. I think the entire gene probably covers.
I have the size. I can show you.
750 kb or something like that.
Oh, I think it's smaller than that. I can find
smaller
it.
than that.
I guess maybe I can just do this instead. Sorry. It doesn't. Yeah, I have the size on there somewhere.
Okay. So the question is, so how fine a resolution do we really have there? In other words, would we know whether or not those QTL are in the promoter region for that gene or in an intron for that gene? I mean, I know you know where they are, but I guess the question is, how fine a resolution do we have, and how much do we know whether or not that's truly, those two QTL you notice there are truly causative? I mean, do we have a 5% chance they're truly causative, or a 95% chance they're truly causative, or like?
It's entirely possible it's a 0% chance too. So
okay, so where are we at in terms of confidence and causality given this level of density of these genotypes?
I think that Troy's work has shown that, and if you listen to Troy's presentation on Tuesday, that even at this 850K density, we don't have nearly as much confidence to make those kinds of conclusions as we do at sequence level. And I think that's one of the things that I'm going to try to do prior to finishing this up, is imputing at least a couple of these chromosomes to sequence level and trying to get more at the, try to refine that signal, right? I don't have any mutations actually within prolactin. I have a ton within that CDKAL1 gene on chromosome 23. So that's kind of why I was part of my motivation for thinking that the prolactin association, it's a possibility that it's just because of linkage with that CDKAL1, those CDKAL1 associations.
Okay, I'll be quiet now. Thank you.
Thank you.
Well, and just to chime in briefly, the other crazy thing that could be going on is all of those associations within CDKAL1 could be tagging an enhancer for prolactin.
Yeah, just hard to tell.
And to follow up on some other questions, I'm not aware of anyone doing hair shedding in dairy.
Yeah, everything that I know about looking at photoperiod stuff, there's been a lot of work on looking at photoperiod in dairy, but it's all been concerned with milk production, not so much with other like seasonal phenotypes like coat shedding. So.
Yeah, it almost makes you wonder if because of the selection for milk, we've cranked up prolactin levels high enough that as soon as the environmental signals come, they switch over that coat and we don't have near as much problems or variation.
Okay, yeah, interesting.
So there's a question from Barry Stevens asking if cattle in Central America shed their hair. And I think that depends on the breed. So there's, like I said, there's differences between coat characteristic is a different thing from coat shedding, but there are some breeds that are like naturally, like Cinepole, that are naturally slick and have that coat characteristic where they have the slick coat all year round. But I think that if you put like a Hereford or any other breed in Central America, they're going to respond to environmental cues and shed their coat in the same way. That answers your question.
Can I chime in on that?
Sorry, what was that?
Can I chime in on that? Yes, they do shed their hair in Central America. You will see them, they do very slicky. They look very slicky. We have brought Red Angus, Black Angus, Charolais, Romagnola back down to Honduras, for example, in the middle of the country. And within a year of being there, that is slick. They look very slicky. They have shed a lot of the hair.
Sophia, do they keep the same slick coat year round because we don't have the same level of seasonality in terms of day length and temperatures?
Yes, it's very rare that you're going to see animals getting hairy throughout the year back down there. Actually, one thing that we see a lot is they get sunburn, and the Charolais and the Holstein, the more white ones, tends to get like peeled in the hips and some other issues just because of the heat. But I have never seen them getting hair back. I have seen them shedding, and it was so fun to me to watch all the seminar because now it all makes sense what I was looking down there a few years back. But yeah.
Appreciate everyone making time to attend today, especially the farmers and ranchers who are collaborators on this project. Again, I'll just echo Harley. you really made this project possible, and we really appreciate you going through that extra effort to collect a new phenotype on your herd.
Yes, thank you so much. And I've gotten to get to know so many people that I wouldn't have otherwise if I hadn't had this level of involvement in it. So very, very glad to have been part of it.
Thank you, guys.